with the swear words from now on. Uh, it's a little bit of a dry subject, I'm afraid. I think so, but I think it's also quite a useful one. It's a mix of stuff I've done much more than I would have liked doing, especially this part of imputation. I did a ton of imputation, much more than I would have ever liked to do. It's not kind of my core business, but something that we kind of have to do nowadays. And have a bit of a chat about where we're going with genomic selection and this data, especially the computational aspects of this. We're at a kind of an interesting crossroads now, where we're generating tons of data. Again, as I said, I'm getting old. And one thing that we have managed to consistently do through all these years is always find some way to generate more data than we can handle. There was a time when the 50K SNP chips were really hard to handle that in our computers. And then nowadays, I can put this on an Excel spreadsheet. Okay, fair enough. So I'll throw a whammy of sequence data at you now, and let's see what you do with that. So we always seem to be running behind this curve, trying to chase up how we're going to deal with the data. And this today is basically what we're going to talk about. But so where are we at? Genomic selection has been awesome. It's really been one of those few transformative things in industry. So we had our blobs, we had our models, then we had genomic selection, genomic prediction, and where we see, this translation from science into adoption into just routine use. And again, this is getting close to 20 years. I think the first early studies were, 2007 or thereabouts, when we started playing around with these SNP chips. And now, pretty much most of the livestock industries are using this rather routinely. So we've really moved from early science to figuring out how to use it, to using it as in routinely. And where are we going with this now? So it's kind of stable. Haven't seen too much great changes in genomic selection recently, but of course, we're always doing new things. I think Guelph is doing a fair bit on the phenotyping side, and I think that's kind of where people are mostly moving towards now, with all the sensors and the bells and the whistles and the AI and the cameras and real-time phenotyping and precision agriculture. So there's a whole lot of scope there to create new phenotypes and real-time phenotypes and throw these into our selection models and for management, etc. So there's a lot of interesting stuff going on that side. There's something going on methods, of course. It's multi and adding stuff. So we're talking about multi-omics, and we're talking about enviromics and other kinds of omics. So it's integrating data sources, data types, and trying to make better prediction models. So there's a fair bit of that as well. And on the more, let's say, genetic side of things, or the genetic data side of things, this is kind of where we're going to get stuck today on, is really about what are we doing now moving forward. Kind of a lot of us has been doing pretty much the same. We sequence reference animals, sequence the key individuals, get the sequence data, get all our historical data, impute the whole thing up to sequence level, and then we do genomic prediction with it. So that's kind of a bit the cookbook right now with the data. We've been doing this, imputing this lower sequence, lower level data, 50K genotypes, even 10K genotypes. I do not recommend that, but people do it. 700K, etc., and bring it all up to sequence level. And now we're moving also into low pass. So it's kind of low coverage sequence data, sparse coverage, and imputing this sparse data into full sequence data, again, using a reference assembly or a reference data set. This is good, this is bad. We'll get there, but above all, we might not have a choice. I know industry is not too fond of it, but we kind of like it with scientists, and the more stuff they throw away, we've kind of find, it's fun. We'll figure out and we'll handle it and we'll do some more fun stuff. Industry is not really enthusiastic about having to deal with sequence data for obvious reasons, but we might not have much of a choice because this is kind of where the service providers are taking us, if we want to or not. The low pass with imputation is actually cheaper than the SNP chips. So they're kind of pushing this harder. So we're going into the scheme-seek data, and this is going to be what we're going to get from now on. You ask for 50K genotypes, it'll give you a VCF with 60 million variants here. Off you go. And that's kind of where we are at, just because in terms of pricing, it's easier. It's more flexible, it's more dynamic, and it's cheaper to do the straight sequencing than to run the SNP chips themselves. It's a bit what happened with the RNA microarrays, if anybody remembers those still. Those are fun. I liked those. They were nice, clear, structured data. And then we went into RNA-seq, which became this big, massive stuff that we have to deal with on a routine basis. It kind of just replaced one. I thought, finally, those RNA arrays will become really cheap. No, they didn't. They just disappeared. And we went to RNA-seq. So that's kind of where we are at now. And wanting to or not, this is the kind of data we're going to be handling. In case you're not familiar with this, I expect most would be, but just a kind of crash course on phasing and imputation. Phasing is basically resolving haplotypes. This is the biggest downside of our genotyping technology, unfortunately. We don't get haplotypes, we get genotypes. So we have to resolve these into haplotypes. We get 0, 1s, and 2s, and we want to know where they fit together and phase this. So that's basically phasing. It's resolving these genotypes into the haplotypes, the resolved haplotypes. Why do we need that? Because we basically need these to be able to do the imputation. So we need to phase them, and then we can impute them. And imputation is basically just filling in the blanks. There's stuff that's missing, we're just completing it. We're filling in missing data, or we're bringing from lower densities to higher densities. This is to fix up randomly missing genotypes. Our off-the-shelf matrix algebra libraries don't like missing data. So if you have a whole lot of NAs, the whole thing collapses, and you can either start reinventing an MKL yourself, or you'll just try to fill up those blanks in some way, shape, or form. Matching different SNP arrays. So this is really what industry does all the time. They have a standard panel, and they align it to the standard panel. We have our version 1, version 2, version 3, 50K, 10K, 20K, 500K, etc. So it's all over the place. When you look at this stuff, there's so many different chips. So what the industry has done is basically, okay, this is our reference panel, and we align everything towards that. Up to higher density from 50K to 700K to sequence, etc. And then, of course, impute the low-pass sequencing. Do not try to use your normal imputation pipelines for low-pass data. It's a rather different animal, so be careful with those. And here's just pictorial in image, in a picture. So imputing the missing genotype. So it's basically what's sparsely randomly missing, just filling in the blanks, imputing across these different arrays, and imputing from low to high. And then you end up with a nice little matrix of genotypes with nothing missing, which will behave the way you hope it would behave. So it just makes our life easier to some extent. But as we move into sequence data, then we start looking into computational problems. Again, this is big. It's large data. Let's say a 30x file. Okay, so this is just 30x coverage. It's not too deep, but it's not too shallow either. So 30x coverage, you get your raw FASTQ files. These pretty looking things here. Most of them are still in these short reads nowadays. I don't think we're going to change from those quite yet. We might soonish, but not quite yet. You end up with about, at the end, 2 times 30 gigs file. So that's kind of your coverage. Your ballpark figure is 1x, 1 gig per pair. So about 1 to 2. That's kind of your raw files. And then the usual bioinformatics gobbledygook. We all have our little recipes, our little what we're going to do. Everybody has a favorite protocols, but it involves always some kind of cleaning and trimming and getting rid of junk, aligning against a reference. This, of course, is going to change very soon now as we move into the pangenome. So we will not have anymore our nice little linear references. We're moving to something else, which is not quite clear yet what that something else is going to be. And then a bit more cleaning, fixing, etc. Then you put all this stuff together and you end up with a GVCF. So you're going from 60 to 37 to 34. And these are kind of your intermediate bits of data. And this is for just one single sample. On this point here, then you call your variance and you end up, let's say, with roughly 8 million, just for references here, about 1 GB. So your 2 kind of points are your raw data. So what do you do with this if you just keep on collecting this forever? And what of the intermediate steps you're going to keep or not? And of course, your final usable data. Just as an aside, my favorite go-to is Plink. I don't use Plink for anything, but I use it for data handling. There's nothing that can handle the sequence level data better than Plink that I know of. It's small, compact, and fast. The Plink 2 is great. Plink 1 really shrinks it. I have hacked a fair bit of these Plink reading and writing methods into my own software. It's just really good. And the new Plink 2 format is awesome in terms of compression and for accessing and subsetting. So basically, my main objective with all this stuff is always to stick it into a PGN format. As soon as I get rid of VCFs, nah. Somebody said once that VCFs is the worst possible format for data storage except every other one. They're probably right. But you can get rid of this stuff by putting it to a PGN. So it's kind of, to go into this place, I would say just Plink it. You can always go back to a VCF from the Plink 2. It now keeps the information, the background information as well. So it's really cool. So I wish I had written this myself. So let's say one animal, fair enough, not so great. When we start moving, let's say just 1,000 animals. That's not so many animals at different coverage. So if you want to keep just the VCF, the FASTQ files, at a 1x coverage, you're looking at 2 terabytes already. So a bit of raw space. And if you have 30x, you're looking at 60 terabytes. So this is just disk space to store raw data that in principle you should not have to use again. And of course, this starts scaling a lot. Even the point one for low pass is going to be still what, 200 gigs? Takes a bit of space. So that's the raw side of things. Just an example with the real data. So 34 million variants, 62,000 samples. You're looking at about 500 gigs for the bed and 8.5 terabytes for VCF. That's a fair chunk of space. It's a whole disk just to store the VCF data. Then, of course, you have to work with it. Memory. So this is on the disk side of things. It really eats up a whole lot of disk. We have now about a peta of storage space there, and that's just for my group, just storing all this junk. On the memory side, on the RAM side, is also the same. If you want to stick this stuff here into RAM, the whole thing, at 2 bits would be your minimal representation, or give or take, that would be 0.75 terabytes. In bytes 3, float 6, and in double 12. So if you would, this is not an intractable problem, but it's a problem that we have to think about and resolve. This is not your OpenR, read the damn thing in and do your Z prime Z and double, off we go, happy days. It won't work. You will not have enough RAM for this kind of thing. So we have to think of ways to parse it, subset it, break it down, etc. So it's a bit more convoluted, which is kind of much nicer when you can just do a Z prime Z and off we go. And for a bit of shock and awe, we can always buy a nice little machine. So we can pay per month 12 terabytes of RAM. You can just about stick it into memory. Can't do anything with it once you get it inside, but it's in memory. And it will cost you $73,000 per month, or you can go for a very economic 3-year plan of $25,000 per month on the cloud systems. So that's kind of your ideas of computational costs to work with this kind of stuff. Computational times also pretty hard. I should probably be not pointing with my hand here. I should probably be using this mouse, shouldn't I? And then the old man can't see anything. All right. So just for the phasing, let's say you split across chromosomes into 1,000 chunks in batches of 50,000 samples. This will take about 16 cores per chunk, 160 gigs of RAM, and 30 mins per chunk. So if you really have the resources, you can get the whole thing phased and sorted out in half an hour. But it will take about 16,000 cores and 160 terabytes of RAM to run this. So you'll need a fairly robust system to do that. We do have that at the university. Again, at the university, not me. So it's the whole university that has that. It's not that I can just access this whole thing. So I can't really go feral, except over Christmas when the jobs that last maximum 7 days finish, and then the whole thing is mine. It's a great time to do imputation, by the way. But anyways, that's on a side. So if we did this on a single machine, it would take about 21 days. Okay, and that's just for the phasing of the stuff, so that we actually have the data before you even start working with the data. None of this is stuff that I like. This is just stuff that has to get done. Okay, so considerations is kind of what we store, raw data, ready to use data, what do we keep, what do we get rid of, intermediate data. The compute requirements. So there's a lot of parallelization and I/O and of processing. So parallel is the key for these things. So it's a whole lot of cores and splitting the jobs. It will always be kept by system limits. So how big is your system? It's how many cores you've got, how much RAM you've got. At some point or other, this is kind of your limits. And we need a lot of smarter programming, approximation, shortcuts, dimensionality, reduction, etc. Daniela Lorenzo, Ignacio Mistal, the group in Georgia, they have been doing a lot of work in this space trying to figure out the best ways to handle these huge data sets. And they've done some really smart work over there on the programming side and on the methodological side as well. And for industry, it's kind of, again, maybe they probably only need the VCFs. And if they send it to the service providers, that's all they're going to get anyways. But what are they going to keep? You know, everything, those 70 to 120 million variants across the entire species, just those within a breed, 10 to 20 million, they already curated in a subset set of 5 to 10. And then what of this do you keep? And then, of course, once you've decided, made these decisions on what you keep and what you get rid of, you're stuck with that. So how do you, later on, if you decide to match across breeds or organizations, the Holsteins say, okay, let's just do it Holstein. So we'll stick with about 3 million SNPs and we're good to go. Then say, maybe it would be cool to do Jerseys and Holstein together, and maybe some Guernsey for good measure. Okay, how do we put this together again? So, you know, all this data is going to be generated. It's going to be big. It's going to be expensive. And we're going to get rid of a lot of it. And what are the consequences of that, or what should we be keeping or not? And of course, how do we revert to the next assembly, to the new assembly? So we have a new bovine assembly, which means re-impute 300,000 animals again. Yay, that should be fun. Okay, so how do we change and remap this stuff? And especially when you go to the pangenomes, there will be a whole lot of consequences on that. And with the historical data and the sequence data, so are we going to impute everything up? Are we going to get 10 million Holstein and impute them all up to sequence? Or are we going to bring it down? I've heard a fair bit of industry saying that I don't care about any of this junk. All I'm going to do is I'll get my subset panel, I'm going to pick them out, and I'm going to delete the rest of the VCF. That's kind of where industry is at now. They just also don't have the capacity to handle this. It's going to be too big. At 10 million animals, at 20 million variants, how are you going to handle and store all this? The breed associations, they have good computational resources, but nothing that gets close to the hundreds of millions of dollars that you have for our new AI systems nowadays. So they can handle it, but not at this kind of scale. And then some basic questions. How good is this imputation anyways? So that's always a question. Is this actually reliable, or is it just a random number generator? And how useful is this? Maybe more important than is it good or bad is what is the value of this stuff? So we'll have a look at a couple of these things first. So first, how good is it? It's a numbers game at the end of the day, okay? So basically imputation phasing, it's pattern matching. The more, if you have this pattern, and I can align it, you're good to go. It's kind of in its most basic format, it's a simple exercise. It's just matching little patterns until they line up. So you have here the haplotypes, and you see the bits that you line up on the lower density ones and fill up the blanks, and happy days. So that's kind of the basic theory behind that. The larger, the better, and that's quite true. The more animals you have in your reference, the better your imputation tends to be. But it is also a relationship game. The more connected this reference and the target are, the better it is. So unrelated animals, if I have 1 million Nilor sequenced and I try to impute Angus, I will still do a very poor job of imputation. Okay, you need numbers and you need relationships. Both go hand in hand. And if I have to pick one, I'll go for the relationships. I'll do an awesome job of phasing and imputing one offspring from 2 parents than from having a huge population that is completely unrelated to that individual. I'll do a better job. Allele frequency is also the same. The more common an allele is, the better. And that is, again, a bit of a tricky thing. An allele that is fixed in a population, okay, you're not going to impute that at all, but if it's fixed, you can't get it wrong, okay? Be careful with imputation of low frequency samples. The correlation will always look good. The accuracy will always look great because you can't get it wrong. If you have an allele frequency of 0.99 and I decide on the genotypes, I can just call everything homozygote. And I will be, look, my concordance is 0.99. It will work, of course. So, and it's also a density game. The higher the density that you start, the better these things go as well. Okay. And now I have a whole bunch of ugly tables here for you guys to look at, but I never give you really numbers of what I'm working with, but here just for an example. So we have some animals at about 10,000 at 50K, 1,700K, and 224 sequence. This is for one breed. Then I have some external reference panel of 6,300 animals from other breeds that are not this breed here. What I had in common was 136 animals with 50K and sequence data. So these are real 50K. We got the SNP genotype, and we also did the same with the sequence. Okay. So that's kind of the background. We wanted to look at a few scenarios. There's a classic one that I used a lot myself in the past, was going from lower density to mid density to sequence in a kind of two-step approach. Or we could go just from the 50K to the sequence. The short version is you're basically better off going from lower to higher nowadays, because this was fine when we had kind of a handful of sequenced animals. So we went from low density to mid density to higher density, because then we got a bit more variation on the intermediate panel. But now if you have a lot of low density and a reasonable size of high density, we might as well just skip over in a single step. Okay. Then, how well does this work using all the sequence, everything except the breed that I'm interested in, only the breed that I'm interested in? And this is a bit of a new one. I think it's the only new stuff here really, is using imputed data for imputation. Haven't seen any other crackpots doing that before, so this was a fun one, and I think we're onto something here. But anyhow, first we can start cheating. So this is cheating where I'm basically imputing myself. I get my 50K and I'm imputing using the data that already has the real sequence. So it should be kind of my best case scenarios. But just comparing this one step and two step. So if you look on the diagonals over here, we did 97, 94, 98, 95, 91, 98. This is a better way of looking at concordance. Don't get the absolute value of concordance. Everything that is very, very common will be very, very right. What you really want to look at is how many of the heterozygotes do actually get right. The rest doesn't really matter so much. So your major allele frequency one will always be the highest. Your other one will be lower, but your heterozygote is the one that you have to worry about. So if you make this kind of table, it gives you a good idea of what is right and wrong in your imputation. So one step, two step, we see that our one step did a little bit better. So we kind of moved on with this for the rest. And here we're doing this a bit more properly. Again, you can always say that there's some bias because you called all the variants together, etc. But anyways, I'm not going to recall everything each time. So over here, using all the external reference and none of my breed of interest, we still have very low imputation accuracies. On the heterozygotes, only 0.56. So it's not a zero, but it's not great either. Okay, and this is a fair bit. If anybody says, I have 6.3 thousand animals in my reference, we'll say, oh, that's good. No, it's not really. Here we included 88 animals, so it's 224 minus 136. So I kept out those 136, and I used only the 88 left over from my breed. And here my accuracy is already much better. My concordance is already much better. So these 88 animals actually do contribute a fair bit. So what about if I just use them? It's much less computing, much less hassle. It's much faster. 88 in relation to 7,000 is so much better. Comes down a bit. Heterozygotes are still even better. I'm doing a better job with heterozygotes using just those than these, because this is just adding allele frequencies that are not really related to the population. But here I'm getting these a bit righter, although I don't have really enough samples to get this properly right. And if I do a bit of round robin, so I didn't have enough samples here, so I kind of use 40, use 190, and keep on rounding through it, and then take the averages, then I get some reasonable numbers. So we can see that relationships matter, and the size of this reference. This kind of just tells us what we already know. But what about if I used those 50,000 that I imputed up, and I tried to see how well I can impute my sequence if I subset it down? That does a pretty good job. So 97, 94, 98. Again, you can argue that there's some level of circularity here because I use the sequence data to impute the 50K, then I bring it down and so on. So it's not completely honest, so to say. But I think we're onto something interesting. I want to explore this a bit more. Haven't had a chance. If one of you guys has a lot of free time, this is something that could be worthwhile to see if we can just use sequence data that we have imputed to impute other populations and keep on adding them. My theory would be that the 700K, I would not use it as an intermediate step. I would impute it up to sequence, and then I would include it in my reference panel, and then go single step. And also, for how long can we do this before we break? So just imputing until the whole thing breaks down. But so that was a bit of a side that I had not seen before. What are we doing with all this? So we now have these ginormous amounts of data at sequence level, all imputed, all sorted. We're happy with the quality of it. Oh, basically what we do, we stick it in our good old mixed models. But if we look at this, okay, you can change the phenotypes, you can change your fixed effects, you can change the number of animals, etc., etc. But given a certain data set, specific data set, the only thing you're ever changing when you're changing your genotypes is this G. Nothing else really matters. So all that you really have to look at whenever you're looking at genotypes is your G. How did your G change in relation to whatever was your standard, your baseline? If this does not change, nothing will change because you get the same numbers. Everything else is the same. So we have to just focus on this here and understand what are we changing here. If we are not changing anything in our G with our sequence data, we're not changing anything at all. And that's kind of the first question. How much is our G changing in relation to our normal 50K panel? Is the G the same or different? If it's different, okay, there's somewhere to go. Otherwise, what's the point of all this? So I can compare these Gs without the breed of interest and with the breed of interest. So this is our real sequence, and this is our imputed sequence. So we're comparing our real data with our imputed data. Here we used all those 7,000 animals except our breed of interest, and there we used everything. Okay. So we did a reasonably good job of imputation. So this is another way of checking imputation. If you have some animals that you can see how well things are working out in your imputation, we generally seed these as well. We kind of get our reference subsetted and including the imputation up to see if we did a good job. And this is without, and we see there's a bit more spread. They kind of align. That is pretty okay. This less okay. There will be some animals that are quite out of it. Another way to look at this is just the principal components plot of the GRM. And we can see the overlap. So they should be perfectly aligned. That would be a perfect imputation. Most of them are pretty close. There are a few that are a bit off. But most of them are kind of aligned. All right, so there are errors. How much do these errors actually affect our GRM? These are different percentages of errors. So we start at 1%, 10%, and now I need my glasses because I can't see the percents. 1, 5, 10, 20, 50, and completely random. Okay, so we see that it's quite robust to errors. Again, it's not exactly comparable. When you're phasing, you have a certain bias in your errors. Here, this is our unbiased errors. We're just randomly changing things. So it's not based on allele frequencies. But you can say that with 1%, won't change anything. 5%, yeah, still okay. 10%, you start to see it splitting out a bit. 20%, even a 50% is kind of still maintains more or less some kind of structure. but only there it really goes away. So it's quite robust to these errors. Probably, the imputation, if we yap too much about, is the low pass imputation high quality or not, they're probably good to go. Don't make too much of a difference, even if the errors are reasonably high. How does it change with different densities? 10K in relation to the sequence. Big differences. That's why I don't like imputing 10K data. I know people do it, and they do it a lot, and I don't like it. I don't think we can get any decent imputation at 10K, but that's just me. Delete this from the recording later on. 50K starts to approximate 100,000. This is kind of where it stabilizes. But at 100K, you're already getting close to where you get with the full sequence data. 500K, 1 million and 5 million. 5 million to 30 million, nothing changes. 1 million, you're also not going to see much changes. So we're kind of on a diminishing returns curve. We have a ton of genotypes, but nothing is really changing in our GRMs. So at some point, there's this sweet spot of how many SNPs we actually need. Maybe 200K would be plenty if we're just going to stick a GRM into our prediction approach. Okay. This is a completely different data set already, but again, just looking at these correlations, the correlations, things are quite similar. 0.98, 0.99, 0.98, so they're quite similar. So you won't see too much changes in your EBVs. How much does this actually help? From theory, we knew it would not be too much, and really it is not too much. So from 50K to 700K, we get about another 1.5%. From 700K to sequence, another 0.6%. This is one example. It's a very closely related breed. The changes are not great. So we do not really gain much through this entire exercise. Okay, so what's the point of all this stuff? Why? Why do I have to spend weeks on a computer doing this? There's some value in multi-breed systems or in small data sets. If you have a very wee, tiny little population with only a few individuals, genotyped, or the actual population is very small, you don't get good prediction accuracies. The sequence data does tend to push this up a little bit. So those red ones over there, it's 150 animals only. We used, where is it? Red on red. We tried to predict the red on 50K, 0.9, red on red, 0.13. So it adds a little bit. No miracles, but it does help a little bit. And the same for crossbreed prediction. With crossbreed prediction, we do see a bit of difference. So the blue one, 3,000, we use that to predict the red, goes from 0.5 to 0.13. So here we're picking up something. If you're going to these multi-breeds, or for one breed that you have information there that you do not, you might have something to leverage here, moving up to sequence data. Here I'm going to skip over to maybe a bit of a different, completely different topic, actually. I've spent half an hour this morning trying to see how I was going to transition from the last slide to this slide. I'm not going to even try. I quit. Okay, this is different. So what we're doing now is we're moving. In the next slide, I go back to where I was. I kind of stuck it in here because I thought it was cool, and then it didn't fit in, but I'm keeping it. So we're moving to pangenomes now. This is our latest and greatest. So two things that we like to do now is pangenomes and T2T. This is where the lab guys are mucking around with. T2T will be great to help us resolve a lot of these haplotypes. So one day, if we really start going for long reads and we have a fair bit of T2T, we will not be bothered anymore with these phasing problems. That will go away. And I hope it does soon. Pangenomes is also conceptually extremely interesting because we are looking at, instead of having a linear reference, we're having this spread out of individuals, and we're looking at the differences between them. This is really cool on a scientific basis, understanding evolution and so on and so on. At the industry level, is how are we going to leverage this, or in practice for genomic prediction, what are we going to do with this? It's kind of cool, but so what? We have a few ideas on this. which I'm hoping to tackle over here on my little sabbatical for the next 6 months, if I stop filling out forms. But this will give us something a bit more evolutionary to work with. It gives us differences. I started doing some work here with the reference panel. So we have our haplotypes, and then tracking these through the pedigrees. All you have to really do is to map out, and this is more for data storage, is to map out which of the haplotypes in the population you're getting and from when to where. So you can essentially collapse individuals into flat files that are very small, because they're basically just a small collection of bits that they have gotten from each of these reference panels. So you say, start with this chromosome from this haplotype, go here, then grab this bit, then grab that bit, and so on. The compression here has been ridiculous. It was simulated data, so I started with the sequence data, then we did a drop down through generations, up to 30 generations. I had the pedigree, so I had no errors in the pedigree, and I have no mutations. So things are, nice and stable. But you get full accuracy in reconstructing this in very, very tiny data sets. And you can think the same about the haplotypes. So right now, I'm still kind of using just this full library of haplotypes of individuals, but you can break this down into smaller haplotypes, which is the minimum number of units that you need to be able to reconstruct things. So in terms of pangenomes, what I'm thinking of, and again, this is just my future science aspect of what I intend, a plan of work, not done work. But what I'm thinking is we can get all these pangenomes and collapse them into a pseudo reference. So I'm going for a longer rather than more common. Some colleagues want to go for the common variants. I'm going for just put everything that is new there and keep on stretching these up. So you don't have any more a reference, you have a pseudo reference. And from this pseudo reference, you have derivations, changes, which essentially, in my view of the world, will be largely deletions or mutations that change things. And that you can collapse into a very tight structure of all the variation and all the variability of the species. And that can be used later on to reconstruct these. So you can really have a small footprint for very large data sets. That's kind of how I'm thinking of it. And this has another benefit, a brilliant idea that I would love to claim is mine, but unfortunately it's from Gustavo de los Campos. So, but I'm probably corrupting it already a little bit with my own things. But it's really a nice idea that instead of having SNP effects, we can have haplotype effects. And these haplotypes, if we go for a structure like this, we're essentially talking about a tree. And this tree is almost an evolutionary tree, where instead of having a SNP effect over here, you can have a lot of effects through the tree. So the first change is a bigger change and has a certain weighting, which is an effect, and then the second, and so on and so forth. So you can be tracking individuals through these trees and adding up the bits of information for these animals all the way through to get a breeding value. And that's, I think it's not exactly what he's doing. That's already me corrupting his idea, but he originally came up with a very tight idea in this space, and he's doing his own thing right now. We have to catch up to see where we are at with this, but I think this has great opportunities. And of course, this naturally suggests what? It suggests a structure for a neural network, where you can kind of hard code your neural network and then start training this neural network through the weightings as you navigate through it with the data. So I think there's a whole lot of interesting stuff that can be explored in this space, but I'm probably dragging on here, so let me move on. And this kind of brings me to, I've had this slide for my life already, probably since I was a young kid. Still haven't solved this. I've spent the last, honestly, 12, 15 years banging on this problem of trying to find better ways to do genomic prediction. Still haven't. But my view is that if you're going to use a GRM, we should use the real GRM, the one that is functional. We should get rid of all the noise, everything that is not part of our trait, remove that and use just what is relevant. Get the weights right, and you should build a GRM that is really informative. So we are looking at genomic selection more as a feature selection problem, trying to get rid of all the noise and all the background rubbish and just use what is relevant. And that should help you. For you guys who are doing GWAS out there, envy it. You'll probably never see one of these in your life. Look, a LOD score of 150. How many of you have seen one of those, huh? Beautiful, isn't it? This, of course, is a very Mendelian trait. It's horns in sheep. But it illustrates the concept. At least in theory, this works. If I use just these SNPs for prediction, I do better than using the full sequence data. So it kind of works on the Mendelian side. We just have to try to scale this into the quantitative side and separating the foreground from the background. And that's really what it is about. This is simulated data now, so nothing real anymore. With simulations, if you have big effects, you can pick up these big effects. And then you should be able to select what you want and include that in the model, remove the background noise, which is the blue. You miss a few small effects, but you can still pick up the big stuff and probably explain most of it. Now, when you have something that is extremely polygenic, say 10,000 informative or causal variants, it's very hard to do anything with this. And that's when GBLUP really rules, because you can't pick up anything better than just fitting the whole thing and hoping for the best. Okay. End of the day, the true GRM, the one that is functional, and the whole GRM is quite different from each other. This one here is using the top 10,000 SNPs. So we basically go there, calculate the SNP effects, and use the 10,000 against 50,000. So it does approximate. There's already a fair bit of differences here, but it does more or less still follow the full GRM. Now, when you use just the real SNPs, which I know, of course, because I created them myself, it's just simulations, we can see that the structure is completely different. And the correlations are quite low. Here's with the truth, and as you start adding SNPs, of course, eventually you converge. So at some point, even if you find, let's say I have 20,000 causal variants of functional informative SNPs that I have to include in my model, you will still be approximating the GRM with the full sequence data. There might be a little bit of biases and variation there, but you're still not in such a great space because you're still kind of capturing the population more than just the effects themselves. My random gratuitous cow. So what have we been doing? This is still not published. It's sitting on my desk for the famous one day when your supervisor will finally submit my paper that I gave him 2 years ago. But we did what tons of people have been doing, and I think this has been the story of everything I've done. I've come up with half a dozen different, I won't say AI methods, but more like machine learning methods. And we've tried different variance formats, created different approaches. I have my kitchen sink approach, which has a combination of 6 different strategies. End of the day, all of these things have never really quite picked up because they sometimes do better, sometimes do worse. That's how it has been in all these papers. Everybody shows that they're doing better, but they never port consistently across others. This is kind of what we have here. So if you look at this, I have a bunch of them for GBLUP, Bayesian, etc. So the more classic stuff all the way to the more ML. In this example here, the overall winner was KNNs, followed by random forest. GBLUP was a bit lower. The worst one was your elastic net. So this is the same population, just where this is oysters actually, and 11 traits. Okay, great. We seem to have some kind of winner here that's better, but it's kind of not so much better because if you look across the 11 traits, once it was Bayesian, twice KNNs, once MLPs, twice neural networks, 3 times random forest, and once gradient boosting. And that's just in the same exact population. If I take it to a different population of the same species with the same traits, I'm probably going to get different results again. So that's kind of where we are at. We still haven't come up with something that is consistently better than GBLUP, which everybody knows, uses, is simple and understandable, and faster. The closest we got were the Bayesian methods, and that's why they saw some level of adoption, because they kind of normally are about as good as the GBLUP, and sometimes better. So that's an option, although they're computationally rather intensive. So I have had this paper there. We'll sit and say, okay, yeah, and yet another paper that says, yeah, sometimes good, sometimes bad. But this is an oyster. Nobody has done it oyster, so we'll just put it out there. But I thought, let's try to do something a little bit different. Let's talk about hyperparameters. This is always one. Whenever you start working with the machine learning, people start, but what are the hyperparameters? How did you optimize it? You have to do the grid search and spend tons and tons of hours looking at these. So we thought, well, at least one thing we could do is look at all these different models and look at the hyperparameters, but not trying to find the optimal hyperparameter, which is normally they say, we did a grid search of blah, 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 and the best parameters were these. We wanted to look at the influence of the hyperparameters. And this is kind of what we've reported in the paper. So the paper has about 10 of these different plots, which is how much do the hyperparameters actually affect my prediction, my outcomes, my accuracies of prediction. And I think this is a little bit interesting. So you can see where you should be focusing on and stuff that is largely relevant. So some parameters, 0.1, 0.2, 0.3, 0.4, or any combination of those will not really do anything, but some of them can have a large effect on your accuracies. I think this is an interesting way of looking at the hyperparameters, not only just running the grid search and waiting until it converges or you run out of time, but trying to understand their influence on the system. If they will hold across different species, etc., I don't know. If each time we run this, we get something different, I still don't know. But right now, this is kind of where we are at this first attempt. And that's the end of phasing and imputation. Since our ML methods are not really delivering great transformational results, that we kind of try other things. And over the years, I've also started to like toys a bit more. A couple of years I gave a talk here and I was all enthused about my portable sequencer and taking it around and sequencing stuff on the fly. And now I got a new toy, which is a hyperspectral camera. So this is my latest and greatest toy. I'm really having fun with this. Although my postdocs are not, because they spend a lot of time taking pictures, but I'm enjoying it. Anyways, the hyperspectral imaging, if you're not familiar with it, it's like an RGB, but at different spectra, different nanometers. So instead of getting just one flat layer, you get many layers. And this has been now it's a big rush also in the imaging side of things. So we're using the hyperspectral imaging to predict anything from milk, composition, methane emissions, diseases, whatever you can think. People are trying to hit with these cameras and trying to find a sense of it. Our main work nowadays is, believe it or not, in strawberries. It's so much cheaper to get a strawberry and cut a strawberry than to buy a steak and measure things on steak. So we did a fair bit of work on strawberries. And my wife works with strawberries as well. So that's also a reason why I got involved with strawberries. But anyways, this is a work we did for identification of milk, of breed composition in milk. Might sound a bit irrelevant. Who the heck cares about if it's a Holstein or a Jersey? Well, the Koreans do. The Koreans care, and they care a lot. They have a perception and a market value for Jersey, which is higher than the Holstein. It's not just based on amount of fat or amount of protein, but an overall market value to it. So they have much more Jersey milk than they have jerseys in Korea. And then they wanted some way to kind of grab a bottle and say, okay, is this Jersey or is this Holstein, or how much of each is it? And that's kind of what we did. So we went to farms, collected from Jerseys, Holsteins, crossbreds. We did different mixes at different ratios of Holstein and Jersey, and did the imaging, tons and tons of imaging. And then basically it's the same. A lot of processes like bioinformatics is now it's a cameramatics. Grab the images, clean it, background correction, foreground correction, normalization, data extraction, summarization, etc. So a bunch of those things until you actually get something usable, which is normally a spectral curve of some sort. We're also looking at different ways of doing this, but something along these lines. That's the variation we had in terms of fat and milk in these populations. It's about 1,000 animals, I think, around that. Not too much differences, but there is some. And just give you the straight summary on the models. So try different models, CNNs, KNNs, PLSDA, support vector machines. That was the best one. So these accuracies are quite reasonable, from 0.91 to 0.97. 0.97 is pretty spot on. Again, don't want to oversell this. We always oversell it. I will not go there. One farm, same food, one camera. how this ports to a different farm, probably these models will decay a lot. If I use a different camera, who knows what's going to happen? Okay, so don't oversell, but the fit is good. Let's say it's a independent fitted data. Okay, so it's not just fitting on yourself, but there is some level of step back here that would be necessary. But by and large, it looks quite promising. Work with strawberry is also looking quite promising, especially for detecting aromatics. So trying to pick up those volatiles of flavor that we use with a mass spec. So we did a mass spec, we're doing the camera, and we're trying to correlate these two. And that has good value because this is expensive and time consuming to do. And we're also looking at the beef side of things to see beef degradation, muscling, etc. and so on. So it's a nice new toy. We're having fun with it. And I'm probably running out of time if I haven't yet. No, still going all right, I guess. Lots of people involved always with these works. Even one from over there, good old Bio Day, still around here. And people who have been working more recently with me is John, who is doing all this imaging, and Umit, who is doing the oyster work. And that's it. Any questions?
Thank you very much for that presentation. I will open the floor to any questions in person or online. We have one in the room. I just had to sneak by you. Sorry. Thank you.
You talked a bit about the computational demand of using sequence data, and I'm wondering if you think that there's going to be an adaptation of the computational capabilities to sequence data, or if we're going to optimize the sequence data to just the aspects of it that we need.
I don't think we'll have much choice on the, we will have to go and optimize the sequence, not so much the computational aspects.
Okay.
I don't think we have the resources for that.
Yeah.
It's all going to get thrown into AI unless you can tag along some way into those structures. We just don't have the money for that. We're always kind of lagging a little bit behind.
Yeah.
Thank you. Any questions? Other questions?
Thank you very much, Cedric. How did you select your 88 SNPs that you used? Was it feature selection or were they very important SNPs? And also, when you're using concordance, what did you, because I think concordance is affected by the allele frequencies.
Yeah.
So why don't you use R squared?
Which R squared? The one from the true and the false, or the one that the software punches out at you?
Yes, the one you estimate directly from the observed.
Yeah. I like the concordance because it's more informative. I get just when I'm looking at the three levels, I get a good sense of what's right and wrong in those little matrices. So I like those matrices for that. I had a look. I always have a look at R squared as well, and the straight correlations, but I just don't find them so visually interesting. But they will tell more or less the same stories. And yes, you're quite right. That's why we look at the heterozygotes.
Were the informative SNPs?
The 88.
You selected from your, if I can remember correctly, you had 0 plus 88 that you used.
Oh yeah, no, those are 88 animals.
Okay, animals. Okay,
animals.
Select animals.
Yeah, it was the 88 animals that I had that I had not in the 50K. They were the independent ones. So I wanted just the independent animals that I had not genotyped and sequenced before, so that I could use this as a reference population.
Sorry, Mike. Flavio, you can go ahead. He's online.
Okay, can you hear me?
Yeah, I do.
So
it goes in the machine.
Part of the body. Sorry that I'm not there, Cedric. I ran out of time. I finished a meeting too late to go there. But before I ask a question, I just like to comment on concordance in R square. I think also depends on the goal of your research. If you ask me, I like to do predictions. I said concordance rate would be a very good idea. But if you are interested in a disease trait and you are concerned about alleles with a low frequency, R square is going to be a better statistics, because with concordance rate, you are going to always find high, right? Concordance, especially for alleles with low frequency.
Yeah. The lower the better.
But the R square is going to tell you for this disease that you are interested in but you are interested to have a good level of linkage equilibrium, right? Is not going to tell you the R square is not or not sorry, quality of the imputation. The R square is going to tell you that is low, and it's really low for the minor allele frequency allele. So you are not going to impute them well. And the concordance rate is going to be always well, right? Because it's the concordance of the genotypes. But if you are interested in rare alleles, then I would look at R square to say how well I'm imputing those.
Yeah, you're thinking in these terms for the filtering, like downstream, I guess.
The filtering?
You're
No, I'm just think about how good is my imputation for the purpose of prediction or studying disease, right? If I like to predict disease, the minor allele, these markers with the low minor allele frequency, they are going to be not well imputed. No, and this is masked by the concordance rate.
Yeah, absolutely.
Yeah. So, but my question is another one. When you showed the results of imputation from different densities of the panels to sequence, so the imputation there was done directly from the panel to the sequence, or was done in like 2 steps, for instance, imputing from 10K to high density and high density to sequence?
Yeah, no, we went straight from the 50K to the sequence for say the proper evaluation. It just worked. Yeah, it just worked a little bit.
Yeah, in all cases was just directly from the panel to the sequence, right?
Not when we were testing out the strategies, when we were kind of doing self-imputation. So I was kind of cheating. I was looking at the animals that I had in the 50K and imputing them up to the sequence, and then just comparing how well they imputed up. And then we saw that the 50K directly to sequence was working better than the 50K to the 700K and then to sequence. So we kind of moved away from the 2-step approach. It's once the reference panel hits a certain threshold, it seems to work better just to impute directly. And if you want to include 700K data, I would impute the 700K data to sequence and then put the 700K imputed in the reference and then impute it all up directly.
Yeah, because all the studies that we did in the past, it was always better to do a 2-step.
Yeah.
When you have a high density.
I know, I did the same. But how many sequenced animals did you have in those days?
Sequenced? Yeah. Not the same as nowadays, right?
Yeah, that's what changed, really. The big change was how much sequence we have available in comparison to what we had before. We were kind of trying to impute up to sequence from 50 animals, 100 animals. Now we have thousands.
Thousands with sequence.
Thousands with sequence.
Yeah.
And then it sorts out your.
Yeah, but the problem is still with the low density, it's hard to sort out the proper haplotypes, right?
Oh yeah. Yep.
Yeah, and then that's why the 2-step would help, because you go from low density to high density or more moderate density, and then to the sequence. The problem was the low density panel to sort out the haplotypes.
Define low density.
Low density. Even
how
50.
low? Huh?
Even 50. Even 50.
50,
right?
yes. If it goes below 50, don't talk to me. But from 50
Yeah,
onwards,
no,
but
50 is, would say low for imputation to sequence is low.
from 50 on, I don't think you have problems. The haplotyping is the tricky bit. That's the lousy bit always. The filling in the blanks is easy once you have the stuff phased out.
Yeah, it's the phasing that is.
Moving from 50K straight to sequence and moving from 50K to 700K, the difference is you're not adding new haplotypes. Once we have enough reference animals in the sequence, you have all those haplotypes, so you resolve the phasing of the haplotypes just as well. It was great when you had the 700K because then you had more haplotypes to try to phase the 50K. But now, once all those haplotypes are in the sequence, you don't need that anymore. They don't add any extra value.
Yeah, but you have the uncertainty of the phase in the 50K because you have a much fewer markers to phase.
Yeah, you do, but the 700K does not help with that. Not more than the sequence.
Well, because there are many.
Unless you have haplotypes that can contribute that are not in the sequence data.
No, it's because there are a lot of markers in common between the 50K and the 700K, right?
So the sequence. How many are you getting? I'm at around 40,000 on the 50K, 43,000.
Yeah, around that, I think 35, 40. Yeah.
So
anyway,
really... we can yap about this later.
Yeah.
Thank you.
Oh, and another maybe it's a question, but a comment. You mentioned to create genome relationships based on markers that are that are kind of really related to the trait. Could be prospective functional markers, right?
Yep.
And that would be better. So I think it was 2008, 2009, we tried to create exactly these panels for each trait in dairy cattle, like production traits, somatic cell score, and so on. So we noticed that with 500, 600 markers, we could predict quite well for milk yield. Instead of under 50K. Then we look at somatic cell score, we need 5,000, and so on for each trait. So when we define the specific panels for the different traits, we end up to have all the 50,000 markers back. And so in practice, it will be much better than to use the whole markers and use the same genome relationship for all traits. So it's just a comment. So that's
no. I know. When I started on this and we put in a project, I kind of finished in this exact same. In those pitfall section of those grant applications, we finished with. By the way, we have all these traits. The chances are that you need all the SNPs anyways, because we will not be able to subset it into a smaller set of SNPs. But that's a fair call. It was exactly that.
Yeah. Okay. Thank you so much for the presentation, Cedric.
Thanks.
Thank you, Flavio. Mike, you have a question?
Well, I can follow.
Okay, yeah. We are a little bit over, but I figured we're already over, but that's okay. So thank you everybody for coming in person and online. There are treats in the back for you guys to have. Next week is a holiday, so we will not be having a CGIL seminar, but we will follow up in 2 weeks.
What's next week?
Good Friday.
Oh,
yeah.
It is. Oh, isn't it?
So thank you, everybody. We will see you in 2 weeks. And thank you, Cedric, for being here today.
Thank you all. Thanks.